From the macro, we're going to move to the micro, and Kymberly Gowdy from East Carolina University will be talk to us about understanding the molecular mechanisms of how air pollution affects susceptibility to pulmonary infections. Do you know how to work that?
I'll figure it out. It's going to try my IT skills, right? So, thank you guys very much for the invitation. This is the first time I've been to one of these conferences, and I'm very struck by the nature of the interdisciplinary discussions. I really appreciate that, and I'm hoping, based on these discussions, we'll be able to add to my talk and understand some more molecular mechanisms going forward of how air pollution can increase susceptibility to pulmonary infections. So, let's see if I can get this. Sorry. Is it the green one? Yes, it is. Thank you. Okay, so as my counterpart, Dr. Cohen, pointed out nicely, we have multiple epidemiological studies that have shown us that there's an association between increasing levels of air pollutants, ambient air pollutants, and infections such as bacterial and viral pneumonia. And this was kind of nicely outlined by Arden Pope's group in 2014, where they showed with the increasing of about 10 micrograms per cubic meter of the amount of particulate matter in the air between 1979 and 1983, as well as 1999 and 2000, that there was an increase in the relative mortality associated with, as I say, hold on a sec. Oop. Sorry. With pneumonia and influenza. So this is one of the sentinel epidemiological studies that toxicologists such as myself refer to. Arden Pope's group has actually recently followed up this study with a really nice paper in the Blue Journal in 2018, where they again showed this association, but they showed that the susceptible population really does seem to be children. So I do think that we need to pay attention to these susceptible populations when we do these studies. Okay, and so not only are we showing this in epidemiological studies, but this is also being able to be recapitulated in laboratory studies. So this was a study by the Floros group at Penn State, and what they did was they actually exposed mice, C57 black 6 mice, to either ozone or filtered air, and then infected them 24 hours after exposure with Klebsiella pneumonia, which is a gram-negative bacteria that can cause community-acquired pneumonia. And what they saw is that the mice that were exposed to ozone had a significant increase in the morbidity associated with Klebsiella pneumonia when compared to their filtered air controls. And this was even shown in some of my doctoral work in Ian Gilmore's lab at the EPA, that when we exposed mice to diesel exhaust and then infected with influenza, that we were actually able to detect higher viral load in the lungs of the mice that were exposed to diesel exhaust versus the air control. So we know these associations, we know the epi studies are out there, and we know these laboratory studies can recapitulate them, but the mechanisms really aren't very clear. So we've been trying to tackle this for about 3 decades now, and I have to say we haven't made much progress, but I'm hopeful that we'll be able to make progress going forward. And so some of the potential mechanisms that are out there of how air pollution exposure can actually increase susceptibility to respiratory infections. I have to admit, Dr. Birnbaum kind of stole my thunder and went over some of these in her presentation, but you're going to hear them again, and hopefully with some repetition, you'll be able to understand them. So we do know that pulmonary inflammation that can be caused by air pollutant can lead to disruption of the barrier function of the lung. And so this can actually not only cause a great amount of cell death in the lung, which can kind of perpetuate the morbidity associated with bacterial and viral pneumonia, but also if you think about sepsis. So sepsis a lot of times starts with pneumonia and then can leak systemically. Well, if the barrier of your lung is not intact, then that is better able for the pathogen to go systemic quite quickly. And so just to kind of give you, this is a really nice figure that I stole from my colleague Rob Ty in one of his book chapters. This is basically what we know so far about how a pulmonary exposure to an air pollutant can cause pulmonary inflammation. And so I'm going to kind of focus my talk on 3 characteristic air pollutants, which are particulate matter. Oops, sorry. Can we go back? The red button? There we go. Thank you. See, I told you this was going to be a training in IT. Now it's schizophrenic. Sorry. Okay. Yeah, I got it. Okay, cool. Thank you. Okay, so I'm going to focus this talk mostly on particulate matter, diesel exhaust particles, and ozone, although there are a variety of different air pollutants that can probably have the same effects. But what we think happens is that you inhale one of these air pollutants, and basically what these air pollutants do is they interact with the lung lining fluid, and they create basically what we call damage-associated molecular patterns, or DAMPs. And this is basically because they're reactive, and so they're going to interact with lipids and proteins, DNA, et cetera, in the lung, and then cause them to be oxidized. And then your body now sees them as foreign. Now, also, a lot of these particle-based air pollutants can actually carry PAMPs, or pattern-associated molecular patterns, with them, such as endotoxin. So therefore, upon inhaling these pollutants, you're being exposed to not only the pathogen-associated molecular patterns, but also the damage-associated molecular patterns. And so then your innate immune response, which has mostly been characterized in the context of pattern recognition receptors, which can be on immune cells as well as epithelial cells, will then recognize these PAMPs and DAMPs as foreign. And this kind of sends off a signal cascade that is mostly driven by NF-kappa B for basically the immune cells in your lungs to produce pro-inflammatory cytokines. And then these pro-inflammatory cytokines then act on the barrier function of the epithelial cells of the lung and basically allow them to become more leaky. And so this not only disrupts against your barrier function, but then you're also going to have a lot of innate immune cells that can come into the lung, which you would think would be able to scavenge some of these, I was going to say, reactive species that are now in the lung. But neutrophils, I feel like they're kind of the big dumb bodyguard of the immune system. So they're really good at what they do. They can scavenge, but they can also create a lot of damage. So neutrophils can be good and bad. But this is kind of the current state of what we know in the context of how an air pollutant can cause pulmonary inflammation. And so there's been some work done that has led us to this paradigm. But the first question is, does an air pollutant actually stimulate the innate immune response? And this has been looking more specifically at toll-like receptors, which are pattern recognition receptors. And this is some data that was from, again, the Thai group at Duke University. And so this is a controlled human ozone exposure. So basically what happens is people come in, they're exposed to filtered air. 24 hours after exposure, they're bronchhed, and they also undergo a BAL. So they get not only the epithelial cells, but also immune cells out of the airspace of the lung. 2 weeks later, then they come back in and they're exposed to 200 parts per billion of ozone. So again, it's a very controlled exposure. And so this is basically the data that they showed looking at the expression of toll-like receptor 4, which is, again, a pattern recognition receptor on the macrophages that they were able to kind of wash out of the airspace. And so ozone, which is an ambient air pollutant, does seem to be stimulating the expression of some of these innate immune receptors, which was, again, this is a sterile lung injury. So not that it's unexpected, but it's somewhat inappropriate. And this has also been recapitulated by Becker et al. In 2005, where they were looking not at ozone this time, but they were looking at human alveolar macrophages exposed to different fractions of particulate matter. So you have your coarse fraction, your fine fraction, your ultrafine fraction. And they were looking, exposure was ex vivo. So basically the macrophages were harvested and then they exposed them in the dish to these different components of PM. And so as you can appreciate, toll-like receptor 2 expression is upregulated with the coarse part of the particulate matter, whereas toll-4 is actually downregulated in the macrophage after exposure. So I think that this kind of lends to the fact that we might have differential stimulation of the innate immune response by different air pollutants, indicating that maybe we need to be studying more mixtures. And so what if we were to actually take these innate immune receptors that are stimulated and kind of take them out of the equation? Let's see what air pollutant can then do to the pulmonary inflammatory response. And this was done by the Laskin group at Rutgers. And basically what they did is they used an OUJ mouse or an HEJ mouse. The HEJ mouse basically has a point mutation in toll-like receptor 4 that renders it inactive. So this is basically like a toll-like receptor 4 deficient mouse. These mice were then exposed to ozone at a variety of different time course. And as you can appreciate, the mice that basically have deficient toll-4 signaling lack an injury response, which is measured by protein in the BAL, which indicates microvascular injury. But then they also have less cells that are coming into the airspace of the lungs. So therefore, they're kind of having a dampened inflammatory response to ozone. And then when they look downstream at some of the transcription factors, you can definitely appreciate that the mice that lack TOL4 signaling have less NF-kappa B activation as well as less pro-inflammatory cytokine production in the airspace. So definitely giving us evidence that some of these innate immune receptors might be driving the air pollution-induced inflammatory response. And so what about an augmented response? So this was, again, done by the Thai group, where they actually did the alveolar macrophages harvested from people that were exposed to either air or ozone. Oh, crud, again, sorry. And so they actually then took the macrophages out and then stimulated them with LPS, which again stimulates TOL4. And as you can appreciate, they are definitely seeing that the previous exposure to the air pollutant definitely augments the innate immune response of these macrophages. And then this was also seen in murine lung tissue with diesel exhaust particles, where diesel exhaust and LPS kind of have a compounding effect with expression of different cytokines and chemokines that can be pro-inflammatory in nature. So all these data kind of taken together definitely does help us understand that the innate immune response definitely does play a significant role in the pulmonary inflammatory response to air pollutants. And so just going into the second, mechanism of how we think air pollution can actually increase susceptibility to infection, and this one's kind of my favorite. We have been able to see that air pollution exposure can actually alter alveolar macrophage, phagocytosis, and intracellular killing. And this is something that my lab is really trying to delve into and better understand the molecular mechanisms. And so if you look at macrophages harvested from the lung, after an air pollutant exposure, you can definitely appreciate that morphologically, they're very different. So this is an air-exposed rat. Oh, crud. And then this is an alveolar macrophage that is harvested from a rat that was exposed to ozone. So just size-wise, they're bigger. They almost look angry. They look foamy. They look pissed off. And then this is actually a slide that I stole from my PhD advisor, Ian Gilmore at the EPA. So this is a differential of cells that we harvested from the lung of a rat exposed to diesel exhaust particles. But look, I mean, these macrophages are loaded with these particles. So, I mean, how are they supposed to do their job after this? If they're morphologically, larger, they've got more foamy appearance to it, or they're even particle laden. And I think this kind of brings us to the point of what we've been talking about today is, what is on these particles? Trace metals are on these particles, endotoxins on these particles. There's a lot that can be associated with these particles. And maybe we're feeding this to our innate immune cells, but we're actually giving, metals that are really important for the survivability of some bacteria. And so therefore, maybe the microbe is then happier in the lung after these different exposures. And so if you look not only phenotypically at these macrophages after air pollution exposure, but also how they can function, this is using phagocytic responses. This is if mice were exposed to either filtered air or ozone. They were then infected with Klebsiella pneumonia 24 hours after. There's definitely an impairment of the phagocytic response. And there does seem to be sex differences, too. So I do think that we need to kind of look at males and females differently in their immune response when we're looking not only at air pollution, but also susceptibility to infection. And so this was done, again, with Klebsiella pneumonia, but then it was also done with Staph aureus, too. So you can definitely see that with either PM exposure or ozone exposure, there is an impairment of the phagocytic response of these macrophages to basically take up bacteria after air pollution exposure. And then, so trying to understand, we know there's a phenomenon that they have different functions. So why is this happening? What are the molecular mechanisms? And this was actually a paper that was done in 1991 by George Jacobs' group at Johns Hopkins. And basically what they were able to show is that prostaglandin, which is a pro-inflammatory lipid mediator, is being produced in the lung. Now, prostaglandin can actually act on macrophages and can actually dampen their phagocytic response. So they asked the question, if we block basically the production of prostaglandin by blocking COX-2 in these mice, are we able to restore the phagocytic capability of these macrophages after ozone exposure? And they did this with endomethacin where they basically were able to slightly restore the phagocytic capability of these alveolar macrophages with endomethacin. But you can definitely see that they probably improved it from 50 to maybe 60%, but we're still not back up to 100% function of these alveolar macrophages that can be seen just with filtered air exposure. But this definitely does help us understand that the lipid metabolism of these immune cells might be altered with air pollution exposure. And so therefore, possibly understanding the metabolism might help us understand what mechanisms are perturbed by air pollution. And so this is something that we've been chasing down with some data from my lab. And specifically, we've been looking at the production of danger-associated molecular patterns, or DAMPs. The one we've been looking at is cell-free hemoglobin, which basically can be produced when red blood cells are lysed, which does happen with air pollution exposure. You can see that your red blood cells are more fragile. And so cell-free hemoglobin, we were able to detect is in the bronchial lavage fluid 24 hours after ozone exposure. So why are there less lysed red blood cells in the lungs? So we're trying to understand the source. But one thing that we've been able to show is that this cell-free hemoglobin can actually alter the alveolar macrophage phagocytosis. So basically these DAMPs are then kind of acting on the lipid membrane of the macrophage, allowing them not to take up the particles. And we've been able to show this with a host defense assay that I've got up there on the right, where if we just take the DAMP and basically expose the mice to the DAMP, and then infect them 24 hours after with Klebsiella pneumonia, you can definitely see that the host defense response is perturbed, and we're getting more bacterial growth in the lung. So I do think that understanding kind of the DAMPs that are being produced in the lung after air pollution exposure is a critical key for us moving this field forward. And so lastly, the last mechanism that I'm going to go through is that we know that air pollution can actually upregulate certain receptors on our cells that pathogens actually use for invasion. So this is something I have to say is probably the least well characterized out of all of the three mechanisms that we know might play a role in how air pollution can increase susceptibility to infection. But I do think that that's something that we definitely need to kind of pour more effort into as immunologists and as toxicologists. And so just to kind of help you understand some of these receptors and what they do, a lot of these are immune receptors that pathogens can actually use to get into cells. And we've specifically been looking at the class of pattern recognition receptors termed scavenger receptors. And so again, these are things that basically recognize patterns that are unique or supposed to be unique to different pathogens. However, we do now know that, certain scavenger receptors such as CD36, SRB1, CD163, and even the low-density lipoprotein receptor are basically reservoirs for how bacteria and viruses can get into our cells. So CD36, TB, can actually use to get into our cells. SRB1 is one of the ways that Hep C can get into our cells. And then CD163, there's actually a porcine-specific virus, PRRS, that actually uses CD163 to get into the cells. And then some other receptors that are not specifically scavenger receptors include ICAM1, which is an adhesion molecule. Rhinoviruses can actually use this receptor to enter into cells. And then even the platelet activating factor receptor is one of the ways that Streptococcus pneumonia, which again is the #1 killer for pneumonia, that's one of the receptors that it can use into the cell. So then, we've kind of been asking the question, are these receptors that bacteria or viruses can use to get into the cells, are they actually upregulated with air pollution exposure? And yes, they are. Not all of them, but some of them definitely are. And so on the left, this is something that was from Urmila Codavanti's group that they just published in Toxi in 2018. And they were looking more at kind of the lipid metabolism of the lung after ozone exposure. But you can definitely appreciate that LDLR, which again is one of the ways that pathogens can get into the cell, is upregulated with ozone exposure. And this is in lung tissue. And then some of the data that we've got, CD163, again, this is, there's a porcine-specific virus that can use this for entry into the cell, but CD163 is upregulated in the lung after ozone exposure. So it's thought that an increase in a lot of these different expression is basically driven by oxidative stress, but we really don't know why. We don't know why basically a non-infectious agent is triggering the upregulation of these different receptors, and I think that that's something that, again, we need to pursue in future studies. So just to summarize what I've shown you thus far, these epi studies and these laboratory studies have definitely shown that air pollution increases susceptibility to pulmonary infection. And I have to say, I've been in this field for about 15 years now, and there's been little movement forward in our knowledge of the mechanism. So for the young investigators out there, this might be a really great area for you to delve into for your future research careers. We're definitely trying to pursue certain aspects of it, but we're only one lab. So some of the different ways that we think air pollution increases pulmonary inflammation as well as decreasing host defense response is through stimulation of these toll-like receptors or these pattern recognition receptors, decreasing alveolar phagocytosis, but again, mechanisms are still at large, and then also increasing the expression of the receptors that some of these pathogens can use to get into the cell. So just for some future directions as well as recommendations, we need more studies. I mean, that paradigm that I showed you by Rob Tai from his book chapter, in the last 10 years, we haven't added a lot to it. So I really think that more studies are needed as well as more funding opportunities for pursuing this work. And then also, there's been tons of epi studies looking at mortality associated with air pollution exposure. What if we went back and we looked at the hospital records and we actually looked at what these people are dying from? I think that might be a really kind of nice complement to some of the already striking mortality studies associated with air pollution exposure. And with that, that's my email address if anyone has any questions or needs a crash course in immunology. That's our laboratory website, and on the left, I have to acknowledge my group. We are working tirelessly, sorry, I can't talk, to try to get you guys more mechanisms so we can better understand how air pollution can increase susceptibility to these different infections. With that, if there's any pressing questions.
Is there time for just a quick one? Yeah. Some pathogens, at least in the gut, when there's an inflammatory response, some pathogens can use the nitric oxide or nitrate pathways as a growth, as an energy source.
Yep.
Is it possible that's a fourth option, that when you've stimulated these innate immune inflammatory pathways, that some airway pathogens can have a growth advantage?
Yes, I mean, that's definitely, I have to say that the pulmonary microvasculature in the lung with ozone exposure or PM exposure is very understudied. We do a lot with the aorta and, the mesentery even, but I feel like the pulmonary vasculature really needs to be better well characterized, and whether or not that's a source of nitric oxide. You could say with nitric oxide, you also get a lot of reactive oxygen species, which are supposed to kill pathogens. But whether or not, and this is something I also would like to challenge the group and future researchers in this area. We're thinking about this from the host, but maybe we should think about this of we providing an environment that a pathogen can grow.
Yeah, that's basically what I'm asking.
Yeah,
And
I was going to say, in the cell-free hemoglobin that we're studying, that's a source of iron. So right there, pathogens love iron. So
great presentation. Thank you.
Thank you.